Good day, everyone. I am Mary Cinco POLANCOS from BSEd Science. Are you guys ready for another exciting discussion? So, alright, let's have first a short prayer. Miss Adelaida, please lead the prayer.
Our loving and generous God, we praise and thank you for the bountiful blessings you have given us. Thank you for giving us a mind that can know and a heart that can love. Thank you for giving us the chance to continue learning amidst the pandemic that had caused a lot of changes in our lives. We are sorry for the times we have failed you. We humbly ask for your forgiveness. Father, help us stay focused on our studies. When learning becomes difficult, grant us the gifts of courage and enthusiasm. Grant us the grace to use our knowledge in making a difference into the lives of the people around us. All this we pray in your mighty name. Amen.
Today, we will be discussing with you another topic in science. This is all about gene mutation. But before that, let us look into the objectives of today's session. At the end of the lesson, you are expected to define the gene mutation, differentiate the types of mutation and its importance, and causes of mutation. To start our lesson, I have an activity for you. So, what are you going to do is to choose the correct answer. You have 10 seconds to answer the question. Alright, who got the correct answer? Okay, very good. Now, let's have some review the past discussion. So who could give me a short review about the past discussion? Okay, Tehome. That's great. Thank you, Tehome. Okay, it's all about genome. We learned that genome is all genetic information of an organism. It consists of nucleotide sequence of DNA.
A mutation can occur spontaneously or be induced by exposure to a chemical or radiation. An agent that causes mutation is called mutagens. Spontaneous mutations are caused due to error of replication and deletion their DNA and may be due to insertion or deletion of bases in the DNA. Insertion or mutation, which extra base pairs is inserted into a new place, while deletion or mutation in which section in DNA is lost or deleted. So going back, base here means nitrogenous bases to exist in two slightly different structure called tautomers. Induced mutation caused by mutagen, which an agent that leads to increase in the frequency of occurrence of mutation, and the process by which mutation is produced will be done as mutagenesis. Examples are radiation and chemicals. Radiation can be ionizing radiation and non-ionizing radiation. Ionizing radiation are X-rays or gamma rays. These are highly energy radiation which can lead to breakage in the DNA structure. They can be single-strand break or double-strand break, while the non-ionizing radiation example is ultraviolet light, which can cause formation of dimers between the edges of bases. For example, thymine dimers due to formation of covalent bond between two thymine bases in DNA caused by ultraviolet light. Now, because of thymine dimers, such DNA is not suitable template for application and it can cause other errors. Then, we move on to the second type, that is chemical mutagen. Chemical mutagen can be of four types. Either it can be base analogs, deaminating agent, alkylating agents, and intercalating agents. Base analogs are two molecules which are structurally similar to the DNA bases. For example, fibromoracil resembles thymine. You can see in this structure, the only difference here is there is a presence of bromine, while there is a methyl group in thymine. Now, this fibromoracil due to structure similarly can form hydrogen bonds with adenine, same as thymine, while on the other hand, its enol form can also form hydrogen bonding with another base, and that is guanine. Now, this mispairing can lead to different errors. Second is the deaminating agent. D means to remove. Aminating refers to a main group. So this is a main group, nitrous acid XS deaminating agent, and it converts adenine to hypoxanthine by changing this amine group into OH group. Similarly, it can change the main group in cytosine to form uracil. Now, changing into these bases can lead to errors or mutation. Next one is alkylating agents. Involve those agents which can add an alkyl group, that is, can transfer alkyl group or substituted alkyl groups to cellular micromolecules by forming covalent bonds.
The types of mutations. Mutation can be classified by whether they remove, alter or add a function, or by exactly how they structurally alter DNA. Point mutation is a change in a single DNA base. It is a transition if a purine replaces a purine, A to G or G to A, or a pyrimidine replaces a pyrimidine, C to T or T to C. It is a transversion if a purine replaces a pyrimidine or vice versa, A or G to T or C. Missense and nonsense mutations. Missense mutations is a mistake in a DNA which result in the wrong amino acid being incorporated into a protein because of change. That single DNA sequence change result in a different amino acid codon which the ribosomes recognizes. Nonsense mutation is a genetic mutation in a DNA sequence that result in a shorter unfinished protein product. Splice site mutations is a genetic mutation that inserts, deletes or changes a number of nucleotides in a specific site at which splicing takes place during the processing of precursor messenger RNA into mature messenger RNA. Example,
Mutations that cause the incorrect splicing of beta-globin mRNA are responsible of some cases of beta thalassemia. Splice site mutation. A point mutation can greatly affect a gene's product if it alters a site where introns are normally removed from the mRNA. It can affect the phenotype if an intron is translated into amino acid, or if an exon is skipped instead of being translated, shortening the protein. Retaining an intron is unusual because most introns have stop codon in all reading frames. However, if a stop codon is not encountered, a retained intron adds basis to the protein coding portion of an mRNA. Deletion and insertion. A deletion mutation is a mistake in a DNA replication process which removes nucleotide from the genome. A deletion mutation can remove a single nucleotide or entire sequence of nucleotides. A deletion mutation that removes 3 or a multiple of 3 bases will not cause a frame shift, but can still alter the phenotype. Pseudogenes and transposons revisited. Pseudogenes is a DNA sequence that is very similar to the sequence of a protein encoding genes. A pseudogene is not translated into protein, although it may be transcribed. The gene may have descended from the original gene sequence, which was duplicated when DNA strand misaligned during meiosis, similar to the situation depicted in the alpha-globin gene. When this happens, a gene and its copy end up right next to each other on the chromosomes. Transposition or jumping genes. Transposition can alter gene function in several ways. They can disrupt the site they jump from shot of transcription of the gene they jump into, or alter the reading frame of their destination if they are not a multiple of 3 bases. Expanding repeats are caused by crucial extension of naturally occurring polymorphic simple tandem DNA sequence within or in the vicinity or different human genes. Myotonic dystrophy is an inherited disease that begins at an earlier age and causes more severe symptoms from one generation to the next. For many years, the anticipation, the worsening of symptoms over generation was thought to be psychological. Myotonic dystrophy is caused by a type of mutation called an expanding triplet repeat. The gene on chromosome 19 has an area rich in repeats of the DNA triplet CTG. A person who does not have myotonic dystrophy usually has from 5 to 37 copies of the repeat. A person with the disorder has from 50 to 1,000 of copies. They have myotonic dystrophy type 2, which caused by an expanding quadruple repeat of CCTG in a gene of chromosomes 3. Affected individuals have more than 100 copies of the repeat compared to the normal figure, than 10 copies. Whether a sequence is random, CGT, CGT, ETG, CET, CAG. Copy number variance. It refers to the genetic traits involving the number of copies of a particular gene present in the genome of an individual. In addition to differing, if only slightly, in our DNA sequence, we also differ in the number of copies of particular DNA. These sequences that vary in number from person to person are called copy numbers variants. Copy number is different from an information than DNA sequence differences. A language metaphor is useful to distinguish point mutation and single nucleotide polymorphism, or SNPs, from CNVs. CNVs may contribute significantly to the differences among us. A, C, and B can range in size from a few DNA bases to millions, and copies may lie next to each other on a chromosomes or tandem, or might be far away, even parts of other chromosomes.
The importance of position. The degree to which alters the phenotype depends upon where in the gene change occurs and how the mutation affects the conformation, activity or abundance of an encoded protein. A mutation that replaces an amino acid with a very similar one would probably not affect the phenotype greatly because it wouldn't substantially change the conformation of the protein. Even substantially, a very different amino acid would not have much effect if the change is in part of the protein not crucial to its function. Globin variants. Because the globin gene mutation were the first to be analyzed in humans, and because some variants are easily detected using electrophoresis, hundreds of globin gene mutations have been known for years. Mutations in these genes can cause anemia with or without sickling, or cause cyanosis, a blue pallor one due to poor oxygen binding. Rarely, a mutation boasts the molecule's affinity for oxygen. Some globin gene variants extend or exert no effect and are those considered clinically silent. Table 12.8. Oddly, hemoglobin S and hemoglobin C are variants that result from mutation that change the sixth amino acid in the beta-globin polypeptide. But in different ways, homozygotes from hemoglobin C are healthy. Both types of homozygotes are resistant to malaria because the unusual hemoglobin alters the shape and surfaces of red blood cells in ways that keep out the parasites that causes the illness. The connection between sickle cell disease and malaria is discussed in chapter 15. An interesting consequence of certain mutation in either the alpha or beta globin genes is hemoglobin M normally. The iron in hemoglobin is in the ferrous form, which means that it has two positive charge. In the hemoglobin M, the mutation stabilizes the ferric form, which has three positive charge and cannot bind oxygen. Fortunately, an enzyme symptoms is usually cyanosis. The condition has been known for more than 200 years in a small town in Japan. Many people there have black mouth because of the cyanosis caused by the faulty hemoglobin. The condition is autosomal dominant. Even more noticeable than people with black mouth are the blue people of troublesome Greek. In blue person disease, the excess oxygen poor hemoglobin causes a dark blue complexion. Carriers may have frighteningly bluish lips and fingernails at birth, which usually improve. This form of methemoglobinemia also affects the Navajo and Eskimos. Treatment is simple. A tablet of methylene blue, a commonly used dye, adds the electron back to methemoglobin, converting into normal hemoglobin. Susceptibility to prion disorders. For the prion gene, as with the globin genes, certain mutations exert extreme effects, while others do not. Recall from chapter 10 that a prion is a protein that assumes both stable and infectious conformations. A prion disease can be inherited, such as fatal familial insomnia, or acquired, such as developing variant Creutzfeldt-Jakob disease from eating beef from a cow that had bovine spongiform encephalopathy. Precise genetic change control the plasticity of the prion protein. The 129th amino acid is particularly important in people who inherit prion disorder. Amino acid 129 is either valine in all copies of protein genotype, causing a form of Creutzfeldt-Jakob disease. These people are homozygous for this small part of the gene. Most people, however, are heterozygous with valine in some prion proteins and methionine in others genotype. Perhaps having two different amino acids at this position enables the proteins to assemble and to carry out their normal function without damaging the brain. Factors that lessen the effects of mutation. Mutation is a natural consequence of DNA's ability to change. This flexibility is essential for evolution because it generates new variants, some of which may resist environmental changes and enable a population or even a species to survive. However, many factors minimize the deleterious effects of mutations on phenotypes. The genetic code protects against mutation. Recall from Chapter 10 that synonymous codons specify the same amino acid. Mutation in the third codon position is effectively silent because the two codons are synonymous. For example, a change from RNA codon CEA to CAG does not alter the designated amino acid. Glutamine, so a protein whose gene contains the change would not change. A change from one codon to a non-synonymous one could affect the phenotype. Another genetic code, nonces, prevents synthesis of very altered proteins. For example, mutation in the second codon position sometimes replaces one amino acid with another that has a similar conformation, minimizing disruption of the protein's conformation. GCC mutate to GGC. For instance, replaces alanine with equally small glycine. A condition mutation affects that phenotype only under certain conditions. This can be protective if an individual avoids the exposure that triggers symptoms. Considered a common variant of the X-linked gene that immature red blood cells use to extract energy from glucose. 100 million people worldwide have G6PD deficiency, which can cause life-threatening hemolytic anemia, but only under rather unusual conditions, eating fava beans or taking certain anti-malarial drugs. What do fava beans, anti-malarial drugs, and dozens of other triggering substances have in common? They stress red blood cells by exposing them to oxidants, chemicals that strip electrons from other compounds. Without the enzyme, the stress bursts the red blood cells. Another protection against mutation occurs in stem cell. When a stem cell divides to yield another stem cell and a progenitor or differentiate cells, the oldest DNA strands segregate with the stem cell, and the most recently replicated DNA strands go to the more specialized daughter cells. This makes sense in organs where stem cells very actively yield specialized daughter cells such as the skin and small intestine. Because mutation occurs when DNA replicates, this skewed distribution of chromosomes tend the DNA most likely to harbor mutations into cells that will soon be shed from a towel rubbed on skin or in a bowel movement, while keeping mutation away from the stem cells that must continually regenerate the tissues.
The nature of mutation. Mutation is a change in a DNA sequence that is present in less than 1% of individuals in a population. Mutate refers to the process of altering DNA sequence. Mutation range in size from alteration of a single DNA base to deletion or duplication of tens, hundreds, thousands, or even millions of bases to missing or extra entire chromosomes. The effects of a mutation vary. Mutations may impair a function, have no effect, or even be beneficial. Loss of function, effect of a mutation when the gene's action changes in some way. Mutation and polymorphism. Geneticists define a mutation as being present in less than 1% of a population, and polymorphism as being present in more than 1% of a population. Not all mutations are harmful, in contrast to their depiction in science fiction. For example, a mutation protects against HIV infection. In germline mutation, the change occurs during the DNA replication that precedes meiosis. Somatic mutation happens during DNA replication before a mitotic cell division. 2. Mutation. Identifying how mutation causes symptoms as clinical application and also reveals the workings of biology. 2 examples of mutations that cause disease. Sickle cell disease, the first genetic illness understood at the molecular level. Ingram identified a tiny mutation responsible for sickle cell disease, a substitution of the amino acid valine for the glutamic acid that is normally the sixth amino acid in the beta-globin polypeptide chain. At the DNA level, the change was even smaller, a CTC to CAC corresponding to RNA codons GAG and GUG. Once a blockage occurs, sickling speeds up and spreads as the oxygen level falls. The result is great pain in the blocked body parts.
DNA repair. Damage to DNA becomes important when the genetic material is replicated. Because the error in sequence is passed on to daughter cells, in response to damage, the cell may die by apoptosis, or it may repair the error. If the cell doesn't die, or the error is not repaired, cancer may result. Fortunately, DNA replication is incredibly accurate. Only 1 in 100 million or so bases is incorrectly incorporated. DNA polymers as well as DNA damage response genes oversee the accuracy of replication. DNA repair consists of cells detecting damage and signaling system in the cell that respond with death or repair. All eukaryotes repair their nuclear DNA, although some species do so more efficiently than others. Mitochondrial DNA cannot repair itself, which accounts for its higher mutation rate. The discovery of DNA repair systems began with observations in the late 1940s that when fungi were exposed to ultraviolet radiation, those cultures later placed nearest a window grow best. DNA repair was inadvertently discovered before the structure of DNA was. The DNA damaging effect of UV radiation and ability of light to correct it was soon observed in a variety of organisms. Types of DNA repair. In man-modern species, 3 types of DNA repairs produce the genetic material for mismatched base pairs. In the first type, enzymes called photolyse absorb energy from visible light and use it to detect the bind to predetermined dimers, then break the extra bonds. This type of repair called photoreactivation enables UV damaged fungi to recover from exposure to sunlight. Humans do not have this type of DNA repair. Second type of DNA repair. Researchers discovered a second type of DNA self-mending in the early 1960s called excision repair in mutant E. coli that were unable to repair UV-induced DNA damage. Humans have 2 types of excision repair. The first type is the nucleotide excision repair, which replaces up to 30 nucleotides and removes error. The result from several types of insults, including exposure to chemical carcinogens, UVB in sunlight, and oxidative damage. The second type is the base excision repair, which replaces 1 to 5 nucleotides at a time but specifically correct errors that result from oxidative damage.